This conference
an
will now be recorded.
Okay, let me switch screens here. Okay, so last time we talked about gene expression, and we talked about specifically the stages of RNA-seq being pre-processing, mapping, quantification, and then together we ran this differential gene expression pipeline. And I think we had a chance to take a look at the different results and talk a little bit about what they mean in terms of fold change, the difference between the means of two groups, and p-value. So p-value meaning that statistically significant change was detected between the two different groups. Then we also did a little overview of these possible 4 steps, so pre-processing, mapping, quantification, and then differential gene expression. And what we want to talk about today more in detail is exploratory data analysis. So exploratory data analysis really is a stage of data analysis that is universally used whenever you approach the data from a statistical perspective. And it's important there to essentially separate signal from the noise, right? That's the main challenge. Because a lot of tests, even t-tests or other tests, they assume that your data contains primarily signal and you don't have a lot of noise. And they also have some assumptions about data distribution and data qualities that we need to address. And so when we talk about exploratory data analysis in the context of RNA-seq, there are several factors to consider here. It has been shown that lab experiments differ greatly between different laboratories as well as different people. So in fact, some have noted that the angle at which you hold the instruments can have an impact on what you are seeing in the data. And when we deal with data that is so detailed and so precise, like gene expression, like RNA-seq, it's very important for us to understand how to address those challenges of noise and technical variation that we want to avoid. So one very useful way to explore your data before you run some kind of analysis is try and understand your data and summarize it in a visual way. One obvious way to do that is map your different characteristics of data visually. For example, one way to do that is using a box plot. So what is a box plot? Box plot shows us different things about our samples. For example, it shows us the maximum value, the minimum value, the median or the middle of our data, as well as quartiles. So if we separate everything into quarters, what do they look like? And then some outliers, right? Which is an important feature of data when you have strange kind of values that don't fit in with how the data looks overall. So when we have this kind of visualization, we can visually compare the difference between our samples. And of course, within each sample, we have quite a number of actual genes and gene expression. So last time, as you remember, we started looking at our data from this perspective, right? So we assembled our data in Excel. And as you remember, we have genes that we already filtered. So we took only genes that are above 6 in their expression value. And we started looking at 3 different things. So we looked at logarithmic scale. How do we normalize the expression numbers using logarithmic scale? And I think that's essentially where we stopped last time. So let me go back to our Excel spreadsheet. Sorry, I think Okay, so our Excel spreadsheet looked something like this. And the first thing that we looked at was the gene expression for two groups, just the raw values, right? So we saw that those values could vary drastically between each other. To visualize that, what does it look like? We can create a box plot right here in Excel. So I think we did that last time. So let me just repeat that really quickly. What I do in Excel is I select all of my data, and of course the columns here are the samples, and then I can insert a box plot called box and whisker in Excel. And this is something that I see here, right? So when I see this visually, what does this tell me? Well, essentially what it tells me is that the majority of my data, right? So if we take a look again at what a box plot really shows, this is what the box plot shows us. Um, it is so this is 50% of the data, right, is the box itself. And then the top whisker is the maximum value, and everything beyond that are outliers. So what you can see in my plot right here is that the majority of the values are actually outliers that we see right here. And then the actual 75% of the data is closer to zero. And look at the highest value here. So the highest value reaches almost 16,000. So obviously this is very abnormal distribution of data, and it will be very difficult for us to learn something about the majority of our data when we have these long tails of data. So what we should do, and let me just show you, I prepared a little bit better looking box plots over here. You can see box plot raw data, right? So I just colored them by groups. This was our normal like, and this was our cloud and low. And you can see the same figure, but basically you see a lot of outliers here and the majority of the data down at the bottom. Now, there are other ways as to how you can see the same thing, but I think a box plot really allows you to visually see several things at the same time. Now, then, as you remember, and by the way, as we're doing this, I want to make sure that you all have the same data right in front of you. So I have two different Excel sheets for this that you can work through together with me, that you're welcome to open on your computer if you want to. And it's in that same Dropbox folder. So you have workbook. This is where we will be doing things step by step. And then we also have this filtered 6 XLS. So let me just copy the Dropbox link and give it to you. And that is where everything is already prepared, so you can actually explore all of this. So what we are trying to see is the difference right now between the raw data and logarithmic scale. And this is really the difference, right? So let's take a look at, after we have converted all of our numbers to logarithmic scale, this is what we are seeing now. A much more interesting visually representation of data, where we see the median and we see the 50%, the middle 50% of our data kind of aligned between 4 and 2, right? And then we have comparable, so more easily comparable maximum values, minimum values right around 0, but not for everything. And then we have another thing that you can see here. So the X represents the mean and the line represents the median. What is the difference between mean and median? So in fact, what it shows us is skewness of data. when the data is skewed, that means that your values are not distributed evenly, they are distributed off to the side. Either to, so in this case, to the upper, so skewness towards upper values. Why is that important for us to notice here? Well, it's important for us to notice in the context of differential expression. Because when we use t-test, we rely on the mean between two groups. And if the mean is different between the two groups, then we will expect some influence of this difference, of this technical difference, on our ability to statistically estimate the value of the difference. And so we will see the impact. We will get actually more differentially expressed genes than really exist in the sample. So to address this problem, what we want to do is use normalization and then compare normalized data instead of comparing data that has this kind of technical variance. And so what would be the normalized data like? Well, the normalized data would align the means and the median to each other. And what we are really trying to do there is normalize each sample to its own noise. So we want to eliminate noise. How do we eliminate noise? We incorporate the values of noise into our values of expression, so we can easily differentiate between noisy gene expression and gene expression that actually matters. Because each sample has its own noise, and if we do that for each sample, we are essentially eliminating the sample noise, and then we are trying to compare signal versus signal. So let me show you on the platform how to do that. So if you go to your account on server t-bio.info, you have a section here that is called utility. So you can click on utility, and then let's upload the raw filtered data. So we have expression genes, FCPM, filter 6. So why are we taking filter 6? Well, primarily, we can either take just data without any filtering. Okay. So you were saying that audio is not good. Let's see how. How is my audio now? Hello? Okay. Okay, well, thanks for pointing that out. Okay. Thanks for pointing it out. Okay, so we will take this filtered 6 data, right? It's in the Dropbox folder that I shared with you. And once it's uploaded to 100%, we can click on Define Groups Check. And here we actually don't need any differential expression, so we can click on Cancel. And now we can click Start. And what you will see here is quantile normalization here under Utilities. So we can open quantile normalization, and let's take a look at what it does. And this is what we saw, what I was just showing you in the box plots, right? So these are box plots. So you have the median value, and you have the 50% of the middle values, and they are aligned, as you can see right here. So we are modifying values according to its own distribution so that they are more easily comparable. And you can, I think, read more about this here in this publication if you are interested to see how other people are using normalization for different challenges in RNA-seq data. So let's just take a look here. So you can see here a whole publication that talks about how normalization, quantile normalization specifically, could be used. Now, there's a question, what other options for different methods of normalization? So there are several methods of normalization that you can use. One of them is just logarithmic scale. So transforming your data to logarithmic scale is already a way of normalizing your data. But let's take a look for a second at quantile normalization. There are quite a number of other methods that you can use. I'm not going to go into detail into what those other methods do, but this is a good overview for those of you that want to try other different options. There's lowest normalization. Again, you can see exactly what it does here. There's quantile normalization right here. And then you can see VSN, or variance stabilizing normalization. And I think, yes, here you even have an example from RNA-seq data. So if you are interested in other options and you want to try them out in R, this is the link right here. Okay, as to the question for when you would want to use it. So like I mentioned here, you want to actually first explore your data before making the decision to use normalization. And you would like to see for specific types of differences, right? So for example, like we saw here, it's quite obvious that you want to normalize this data in some way. But before you use a specific method of normalization, you want to look at your values in a way that would be more clear what parameters of those values are off or not normal. And we did that by just transforming it to logarithmic scale, so we actually can see the box plots. Now, if you want to look at specifics of values of this, there's another thing that you can do called summary statistics. And summary statistics is also something that you can do in Excel or in R. Here, the example is in Excel. So let's take a look at what we see here. We get several types of parameters, statistical parameters, about our data. So we have the mean, standard error, median, mode, etc. So we have all of these different things. And so, for example, we can take a look at the median, and we can take a look at skewness, right? So I was talking about skewness before. So here you have the median, and here you have the skewness. So by median, you can see that just transforming your values into logarithmic scale, you actually eliminate a lot of the differences. So if right now we leave out the raw data, and let's just change the size of this. So maybe we'll keep it at 5, or maybe 3.5. So the median values are really comparable already for logarithmic scale, right? Now, the second parameter of skewness, you can see that there's a difference between the groups in skewness. And this is very apparent when you look at logarithmic scale versus quantile normalized, right? So here you can see the skewness is higher for just the group of normal-like samples. As opposed, so because I see this big difference, I am going to be expecting it to influence my my ability to use tests like t-test. So that's when I'm becoming concerned, and I'm starting to think, how do I actually address some of this variation? Let's take a look at some of these other things that we have here, right? So for example, we can just take a look at the mean. So this is the raw data. So let's just take a look at what's here. So this is the mean for raw data. So let me do two things. First of all, I will add the names of samples. And the second thing, so this was raw data, and let's add here logarithmic scale. So that would be right here, right? So I will take the mean. And so you can see that the mean is practically difficult to see here compared to these high values over here. So let's add here real quick also quantile normalize. So actually, okay, this is going to be okay. So let's take a look at maybe without the raw data. Okay, so what I want to do is going to be ln scale, and I want to use mean values and the names. Okay, so even here, what you can see is that on average, just technically speaking, right, we are going to be comparing the mean between the two in t-test, and it's higher for normal like as opposed to cloud and low. And because of that, right, so what is a mean? Mean means that we take all of our values and we find the middle of them. So we actually divided by the number of genes that we have. And what we are expecting, obviously, in two different conditions, we're not expecting all of the gene expression to be significantly lower. We are expecting various genes to change their expression. So maybe some will be higher, some will be lower, and the sum, those groups, would be different between two groups. Not overall, all of our gene expression to be suppressed. And so because of this, we now want to normalize the data. That would be the rationale for trying to do that. Does that make sense, Malik? Does that make sense on why we are doing normalization? Okay. All right, so these are just some examples. Just briefly, how do you get these summary statistics? It's very simple in Excel. So in fact, in Excel, you have the option to enable data analysis. And if you click on that, you have here different things that you can do. So for example, one of the things is you can do descriptive statistics. And if you do descriptive statistics and you have labels in first row, and then you put in a new worksheet and you do want summary statistics, right? So if you do that, it's going to generate, okay, so I didn't do the input range. So I can do summary statistics on normal data. And if I click on OK, it is actually going to generate a new, I went to the other screen, but you see it generated all of this information. Now, this table is a little bit difficult for me to visually look at. So what I would do is I would just select all of these names of columns and paste them here. And paste them here. And then what I can do is I can actually delete all of the repeats between the row names. And now I will have a table that I can easily work with, right? So this was raw data, descriptive statistics. Okay, so this is some idea on when to use normalization. So let's, we don't need this because I already have that in here. Let's go back to the platform and complete our pipeline. So here we have two selections. So we can already convert to normal logarithm scale all of our data. We want that. And then we can use a threshold. So because you saw that the majority of the values are very low, we actually want to normalize only the top. So we want to leave everything that is below certain value without touching it, right? So here we can use, for example, 10 or something like this, low values, and then we can click on end, and let's do QN, and this will be QN of 10, cell line expression data. Let's run this pipeline. Okay, now you can all try and do this on your own and see. So I can choose here different options. I can filter by 6. Okay, how to select threshold value. So actually, again, going back to these box plots, right? So what it will do is you see that these are logarithmic scale numbers, right? So all of my data is under 10, but the majority of it is under 6, right? So that's kind of right below the maximum value. So what I actually should do is select 6, or I can select 5, because there's a certain, you know, data variability under this value that I should leave alone. And so, you know, what you have is actually two different options. So let me just show you here once again what are the options. Again, remember that I select logarithmic scale, so even though I have filter of 6, I'm not relying on the raw values. I'm using here the values after logarithmic scale. So let's just take a look at what other options I have here. So I have okay, so yeah, if you read the description here, you actually have a good idea of what we are doing here. But as you saw in the box plot, so I can select that. But you basically have to look at it in the way that we did right now. Okay. Sorry about the audio issues. I don't know why they keep coming. Maybe it's when I upload files, there's something with the connection. So, you know, we can play around with this with the different parameters and see what the results would be. Okay. All right. So here we are running several pipelines. We'll take a look at the results. So what would happen if we take all? If we leave our data as 0. So if we leave that threshold at 0, what will happen is that all of our data, as you can see, is going to be exactly the same, right? So what would happen if we did it as 0 is that everything is perfectly aligned, right? And that's not a very good thing. So if we align everything perfectly, we are going to be adjusting it maybe a little bit too much. But this is an example. Let's take a look at what would happen. What would be the impact on, for example, differential gene expression? So let's take a look here at first of all, how do we see the change? So you see that everything is going to be perfectly aligned. So median, skewness is going to be totally fixed. So everything will be fixed, but maybe a little bit too much, right? So we will take a look here in a little bit what will happen after we use thresholds. But also what we can look at is how this will change the number of differentially expressed genes. So if differentially expressed genes are going to be basically an adjusted p-value, which is statistical significance of the difference, and log fold change, right? And so based on these two factors, I can calculate how many differentially expressed genes would I have. So if I just use raw data without any transformation, I will get approximately 194 genes. If I do logarithmic scale, that number would reduce to 133. And then if I do quantile normalization without any threshold, I will get down to 32 differential expressed genes, right? So the change is quite dramatic, right? You can see how many of raw differentially expressed genes I have, how many of logarithmic scale, and then how many after quantile normalization. And here I should mention that it's quantile normalization after or threshold 0. So let's just clarify that. Okay, now let's take a look at what will happen when we do, for example, 5 or 6. So my pipeline has finished. I can select which one I want. I already have the 6 here in the folder. So if I take a look at threshold, right? Okay, threshold of 5. Okay, so I can open this in Excel. So here's my data. And let's just make sure that the samples are in the same order. Okay, so 687, 688, 720, 721, 634, 697, 703, 714, 716, 26, and 36. So it looks like, yes, it's the same. So let's take a look. Let's just copy. Again, let's just also make sure how many do I have? 9,084. So let's take a look at how many do I have here in total? 9,084. So it seems like I can just copy this and I can put it right here. Okay, and let's just insert copied cells. Okay, and let's do the same thing like we did before. So as what we did was add a column here that says normalized. So we can say normalized threshold 0 and copy this, and then we can just paste it here. And this will be normalized threshold 5. Okay, and now what we can do is we can copy these as well. Just copy them here. Let's move this over a little bit. And we can also copy this. Okay, let's just make sure that we did everything correctly. So first of all, let's just make sure that it looks the same visually. I think it's confusing when we see differences just visually. And now let's just make sure that the values are correct. And let's take a look at summary statistics for this, right? So what we can do is a box plot. So let's actually duplicate this normalized. So let's duplicate this. And so here we will call this one normalized 0. and this one we'll call it normalized 5. And now let's just change the data. And so we have all of our samples. So we can go ahead and do select range and then go to let's keep going. Okay. And this would be our range right here. Okay, so what do we see here? What we see, let's adjust this to go to 10. And what we can do is we can just color them really quickly so that they are the same. And so we'll have, and then we'll have a border. So all of these are normal like. Let me see real quick. Oh, okay, so these would be green. Sorry about that. Let's do that with light green. And the first 5 are going to be like this. And then the rest are going to be light yellow. Okay, let me know if you have any questions in the meantime. But just want to make sure that everybody can make the same types of charts and really see for yourself what we are looking at. Again, we'll move on to the next example here in a little bit. So what do we see? Sorry about this. What do we see here? We see here that the normalization, oh, okay, and what we also don't need to see is the inner points, and we want to see the mean line right here, right? So we don't want to see the inner points. So as you remember, we were looking at skewness, and the skewness was visible when we saw the difference between the mean and the median. So let's take a look at what we see here. And so I'm going to remove the inner points so that they don't interfere with what we're doing. Okay, so what do we see? Now we see a much more comparable picture, right? But it's not too adjusted, so it's not too bad. And what we can also see, so let's just change this quantile normalization threshold 5. And let's put here after quantile normalization threshold 0. So this is maybe too much, and then we normalize them and we see that this still has some variability. And of course here let's just quickly see mean marker here as well. And let's see what we will see in terms of how the mean varies from the median. Okay, so see, here it's going to be exactly the same. And that's a little bit suspicious, right? You actually don't expect everything to be exactly the same. Then what would be the purpose of really, you know, evaluating global changes? But now we address that problem by introducing a threshold, and now we have some variability. Let's see how does this variability compared to logarithmic scale. So what changed? First of all, the size of the 50% box changed, right? Now it's more comparable. Here it's, this is very small, this is kind of larger, right? And so what we want to do is align the middles. And once the middles are aligned, we can now compare everything in a standardized scale, so to say, right? And then we saw that here, everything was too perfect, and then here we can see that the sizes are the same, comparable. So it's kind of like comparing apples to apples and oranges to oranges instead of apples to oranges. Okay, so now that we have this, let's just quickly add some summary statistics and see what happens here, because we want to address that issue of skewness. So let's go back to our table right here. Okay, so this is normalized 5, and what we want to do is data analysis and descriptive statistics, and the input range is going to be for this new portion right here. And we want to have labels in first row, and let's select an output range. So we want to go to summary statistics and have an output range right here. Summary statistics, okay. And let's see what it does, what it shows us. Okay, so as you remember, we want to take all of these and move them over. And now let's just copy all of this so we can remove those extra rows. Okay, so let's remove these extra rows, or extra columns, sorry. Okay, and now we have the same kind of a table. We can copy it back here. So this is going to be quantile normalized. So this would be quantile normalized threshold 0, and this would be quantile normalized threshold 5. Okay, so now let's see what we have for median. So let's just highlight this one. And for skewness right here. And let's see how those compare. So skewness, let's add it here. So let's add quantile. So actually, this is right here. And the values are right here. So what do we see here? Looks like I got some misalignment here. So let's take a look here really quickly. What did I do wrong? Right. Okay. Looks like we actually don't need the first one, so just these. Okay. All right. So, and raw data, something happened here with raw data. So let's select data here real quick. Raw data. Raw data, maybe I changed the wrong one. So skewness for raw data and skewness for smooth data. Right. Okay. All right. So this is what we see. And what we want to do is now let's remove this, and we'll leave just the ones without the raw data so we can more easily compare. So let's do data, and we'll remove raw data from here. Okay, so now let's change this, and we said it's about 3.5 as the maximum. Okay, even lower than that. Sorry, maybe 1. Okay, so now we see that we had originally in the logarithmic scale just very high skewness of logarithmic scale for normal-like versus cloud and low. The red one, which was quantile normalized threshold 0, made them exactly the same. And quantile normalized threshold 5 evened them out, but not exactly, did not make them exactly the same. Okay, and now let's take a look at what this shows us in addition to what we saw before. So I guess what you're starting to see here is how normalization and transformation to logarithmic scale can help us see the real values that are more comparable. Now, since we don't have a lot of time, I want to move on forward and tell you talk about 2 more methods that give us a different perspective on the data, not a statistical perspective per se of qualities of data, but it talks about how to visualize this multidimensional data and understand a little bit the structures that we expect to see. So what is the main structure that we expect to see? Well, the main structure that we're expecting to see is some difference between the 2 groups. We have, on one hand, the normal-like samples. On the other hand, we have the normal-like. So normal-like and cloud and low, sorry. And we expect some separation between those 2 groups of samples. So one way to do that, another method of looking at this, is doing a scatter plot. And a scatter plot will help us identify in a 2-dimensional plane how these samples separate by their expression values. The problem is that we have too many values, right? So if we, for example, go back to our table here, and arbitrarily we just say, you know what, these are the first 2 genes, how do the samples separate by 2 genes, right? So here I can easily insert a scatter plot, and I will see something like this. So I have 2 genes, right? Okay, so actually what I want is not this. What I want to do is just select the first 2 genes. One will be X and one will be Y. And now I want to actually separate them into 2 series. So the first series is going to be normal-like, and that would be these 5. And these are going to be the Y values for these first 5. And then I will add for cloud and low. And I will do the same thing, but 1, 2, 3, 4, 5 for these 2. So this and this. I see some separation, right? So I see separation between normal-like and cloud and low. But I only selected these first 2 genes arbitrarily, right? So I don't really know, is this representative of all of my data, all of the data that I have about my observations? Observations here are samples. So what I want to do is use a technique called principal component analysis. So let me explain to you how principal component analysis works. Okay, so we talked about normalization, and we talked about the difference in gene expression, and we talked about summary statistics. Right, and you will have this presentation or this video recording in the end as well. And hopefully you will understand how to utilize these normalization techniques for your next project. Okay, now we will talk about principal component analysis. So let's try and understand what this is. First of all, principal component analysis is a very good summary technique to visualize all of your data without losing the data, without just selecting two genes and discarding the rest, but at the same time transforming it into an easily visible two-dimensional plot or a three-dimensional plot. So first of all, what is this? As you saw, we selected a couple of genes, right? So we have two dimensions, X and Y, two axes, two dimensions, gene 1 and gene 2. And we can plot our values just like what you saw now, right? So we have some group of samples and another group of samples, and their X and Y values are coming from expression of one gene and the second gene. And now we call all of our genes n dimensions. So in our case, n equals to 9,000 and something. We can take three dimensions, right? So the first three genes and visualize them in a three-dimensional space. But obviously with 9,000 dimensions, that's impossible to visualize. And so now what we can do is we can try and project principal components, which are going to be vectors, so lines in this multidimensional space, in this space of 9,000 and something genes that show the variance. Okay? What is variance? Well, what we are trying to see in our data, one thing that we are trying to see is the variability of the numbers within each group. Just like we were looking at box plots, right? And we were looking at the skewness and distribution and other things. Here we are really looking at variance. And we are trying to capture the representation of variance between those different samples, for each sample separately. What is variance? Well, this is low variance, an example of low variance, where they are grouped closely together. High variance is when they are going to be spread out. So if we try and capture the variance of the samples right here, we will try to find a line that goes through the most variance, most variability of this data set. And that would be principal component 1. And then we will find a perpendicular line to the principal component 1 and project it in the second most variant direction. And as a result, we will get principal component 1 and 2, and we can have 3, 4, 5, etc. And then we will replace our X and Y with those principal components. So we will do a projection of all the values that we have on these principal components. And so as a result of principal component analysis, what you will get is a scatter plot that projects sample values, right? So we are looking, dots represent samples here, onto principal components. An example, we have all of these genes as our dimensions and all of our samples. And what we will do is do this simple pipeline that will help us explore the data variance and understand our data from the perspective of principal components. So let's go and do that really quickly. Let's go to areas of analysis and do unsupervised analysis under data mining. And now what we can do is let's add the file that we just got from normalizing the data. Okay, let's take this one. Continue. And now we change a little bit the name of the button itself. Instead of PCA draw, it's PCA R library. And I will show you that actually you can download the R script here as well at the end. So we will try and do 3 principal components is what we are interested in. We can choose to scale the data if we wish to, center it so that it's more adjusted on the graph, and transpose the data because we have genes as our rows and samples as our columns. And typically PCA actually imports it the other way around. So now we want to see the way our samples are positioned in this multidimensional space. So let's call this PCA cell lines filtered 6. Okay, and let's run this pipeline. Now you can do the same thing. Let's take a look at what are we going to expect to find. So, well, what we are going to see, and we'll download the file here in a second, is something that looks like this, where this is, of course, PCA of all of the samples, but now we're just looking at cloud and low versus normal-like. So we see some separation here of these different samples, right? And these dots are projected on PC1 and PC2. Here we have another example. So again, these are all the samples, and I'm happy to give you that data, and I will in future lectures, just so that we can understand better how to deal with more variability between the different subgroups of cancer, right? That's our goal in the end. And so here what you can see is how PCA analysis, so principal component analysis on 7,000 genes, shows us different subtypes of breast cancer studied in the cell line project, and where we can see, as you can see, this is luminal on one side, and it's separated from the rest. But even with the rest, we can see some separation between basal and then normal-like and cloud and low here on top. Now, as you remember from our first lecture, when we talked about the project itself, we can actually look at PAM50. PAM50 is a panel of 35 genes plus some housekeeping genes that are added onto it. And according to PAM50, you can see a separation. Interestingly, you can see normal-like and basal kind of together, which actually biologically makes sense. And then you can see cloud and low over here and luminal over here, right? So this kind of gives you an insight on how clinically can gene expression be used to understand the differences between different subtypes of breast cancer. Okay, so this is how you would separate the different groups, right? And we can also label them. So let's take a look at our pipeline and see. It should have finished by now. All right, it finished. So let's take a look at the result. What do we have here? So briefly, we have the text file on some statistics of PCA, principal component analysis. We have all of the results zipped. We have the plot that we were just looking at. Let's just download that. But also we have here the R package. And next time I'll talk a little bit about this, how you can take this basic script and improve it to demonstrate some additional things on the graph itself. And then you have the PCA table. So let's quickly take a look at PCA. What did we get in our downloaded file? So we have this kind of a file, okay, where we can see the samples. And of course, they are not marked, right? So right now, we can just guess what they are. But I will quickly show you now how you can take and modify your table to make this appear as differences between groups, but also how you can just quickly do the same thing in Excel. So here I can download the table, and I can open it in Excel here. And this is what it looks like. So here are my objects, my observations, or my samples. So what are they? They are in the same order, so I can just go to 634, and these are going to be my normal-like, and these are going to be my cloud and low. So I'll just highlight them as yellow. And now what I can do is I can insert here a scatter plot, and that's really all that package in R does, is it runs principal component analysis, generates these values, and then creates a scatter plot. And so now I'm going to, so this series is called normal-like, and then I have a cloud and low. So for cloud and low, this is going to be my X values, and these are going to be my Y values. And now I have a principal component analysis result for the samples that I have, right? So now I can add here a little explanation or the legend. And now I can see that these are normal-like and these are cloud and low, and they are clearly separated. And so what's interesting for this is that we can see the difference between these groups of samples based on all of the data that we have, all of the observations, all of the gene expression values. And that is a unique thing about principal component analysis that gives us some insight into how they are separated. That is all that I have time to cover today. Next time, we will talk about several clustering methods and how we can use clustering to understand structures, subgroups in our data. And we will also try and work with all of the subtypes of breast cancer to really understand the differences between breast cancer subtypes in our full project and full data set. We'll also do a little review of the data set and its significance. Before we leave, do you have any questions or any concerns about how we ran the pipeline or how we discussed the data that we saw today? You can either unmute yourself or okay, so PCA is not really verified by clustering. It's a different approach from clustering, but it does show similar things. So for example, we see here some major separation into two subgroups. So we probably expect there to be at least two clusters, right? And so in that sense, clustering can give us additional insight, but one of the unique features about clustering is that it will give us a little more detail, but that detail is actually a little bit up to us on how we interpret it. Any other questions? All right, great. Thank you, everyone, and hope to see you next time. Take care.
Thank you.